Today's presentation is a discussion of an application for the In-Cell Western Assay on RNAi screens developed by our speaker, Dr. Greg Hoffman, in the laboratory of Dr. John Blennis at Harvard Medical School. Joining Greg today as panelists are Nate Moriarty from Harvard and Peter Johnson, a field application scientist for Lycor Biosciences. Greg's talk is titled, Development of an In-Cell Western-based HTS screening method for the quantitative analysis of mTOR-C1 signaling. We are proud to turn the floor over to Dr. Gregory Hoffman, postdoctoral research fellow at Harvard Medical School. Greg?
Thanks, Ashley. Good afternoon, and welcome to the laboratory of Dr. John Blennis here at Harvard Medical School. Today, I'd like to talk to you about the work that I've been doing in the Blennis Lab on the development of an In-Cell Western-based high-throughput screening assay for the quantitative analysis of mTORC1 signaling using the Lycor Arius infrared scanner. This work was carried out in collaboration with Nate Moriarty and Carolyn Chamu in the Harvard Institutes of Chemistry and Chemical Biology, the high-throughput screening facility here at the medical school. Before I describe the details of our In-Cell Western assay, I'd like to give you a brief introduction to the mTORC1 signaling pathway and describe the rationale for our high-throughput screening projects. The Blennis Lab is interested in understanding the molecular basis of cell growth control by the mTOR signaling network. The study of mTOR began with the identification of the natural product rapamycin, which was isolated based on its potent antifungal properties and has since been shown to dramatically inhibit the growth of nearly all eukaryotic cells. The protein kinase mTOR was identified as the cellular target of rapamycin, and the allosteric inhibition of mTOR kinase activity is now known to be responsible for the growth suppressive effects of rapamycin. Rapamycin is currently in use clinically as an immunosuppressive agent for transplant patients and is being tested in clinical trials for its efficacy in treating a variety of cancers and other tumor syndromes associated with misregulated cell growth. mTOR exists in two functionally distinct complexes, referred to as the mTORC1 and mTORC2 complexes. The mTORC1 complex, which is the focus of the work that I will describe to you today, is comprised of the mTOR kinase bound to two additional proteins, Raptor and LST8, as shown on this slide. mTORC1 lies at the center of a complex signaling network that is responsible for maintaining cellular homeostasis. Specifically, mTORC1 integrates multiple upstream signals, including growth factor availability, cellular energy status, and the availability of amino acids, and in turn controls key processes that drive cell growth, such as macromolecule biosynthesis and glycolytic metabolism. Given its important role in cell growth regulation, there's been a great deal of interest in understanding the molecular details of the events controlling mTORC1 activity, and our screening efforts are aimed at identifying novel components of these upstream signals responsible for regulating mTORC1 signaling. This next slide shows a more detailed view of the progress made over the past few years in understanding the molecular details of mTORC1 regulation. Growth factor and energetic control of mTORC1 involves regulation of the TSE1-TSE2 complex, which acts as a GTPase activating protein, or GAP, for Rheb. In its activated GTP-bound form, the Rheb GTPase acts as a direct activator of mTORC1 kinase activity. The mechanism of growth factor-mediated signaling to mTORC1 is now relatively well characterized. Activation of kinases including AKT and RISC in response to growth factor stimulation leads to the phosphorylation and inhibition of the TSE1-TSE2 complex, increasing Rheb GTP levels, which in turn activates mTORC1. Conversely, activation of AMP kinase in response to a decrease in the cellular ATP-AMP ratio leads to phosphorylation and stabilization of the TSE1-TSE2 complex, downregulating Rheb and leading to inhibition of mTORC1. One question that remains with regard to Rheb activation is the potential existence of a putative guanine nucleotide exchange factor, or GEF, that may play an important role in stimulating Rheb activity. At this point, no Rheb-GEF has yet to be identified. In contrast to the growth factor and energy sensing pathways, mTORC1 regulation by amino acids is less well characterized. Recent work from Lowe and Murphy at Novartis has shown that a glutamine-leucine cotransporter at the plasma membrane is critical for allowing leucine into the cell, a key step in mTORC1 activation, while David Sabatini at the Whitehead has identified the RAG GTPases as key regulators of mTORC1 localization in response to amino acid stimulation. However, the intervening steps in amino acid signaling between this glutamine-leucine cotransporter and the RAG complexes are not yet well defined. Specifically for the assays described here, we use an antibody that recognizes S6 when phosphorylated at the 235-236 sites. To address some of these unresolved questions, we set out to develop a high-throughput, cell-based assay capable of quantitatively measuring mTORC1 activity, with the goal of applying this assay to both small molecule and RNAi-based screens. The assay we developed relies on measurement of RPS6 phosphorylation as an endpoint for the activation state of the mTORC1 pathway. As shown in the schematic on the left-hand side of this slide, mTORC1 activation leads to the phosphorylation and activation of S6 kinase, which in turn phosphorylates ribosomal protein S6 on multiple serine and threonine residues. S6 phosphorylation may play a role in regulation of protein translation downstream of mTORC1, but for our purposes, it provides a convenient and reliable readout for the activation state of the mTORC1 network, owing to the availability of high-quality phospho-specific antibodies recognizing these sites. Specifically, for the assays described here, we use an antibody that recognizes S6 when phosphorylated at the 235-236 sites. The characterization of this antibody in HeLa cells is shown on the right. Immunofluorescence analysis is shown in panel B. Cells were treated with DMSO, the mTORC1 inhibitor rapamycin, the dual PI3 kinase mTOR inhibitor LY294002, or the MEK inhibitor UO126. Cells were fixed and stained for S6 phosphorylation in green. Actin filaments were also stained to help visualize the cell morphology and are shown in red. As you can see, rapamycin and the LY compound potently inhibit the phospho-S6 staining. Importantly, UO126 had no effect on mTORC1 signaling, demonstrating that the ERK-MAP kinase pathway does not play a significant role in regulating S6 phosphorylation under these conditions. A similar experiment was performed by Western blot analysis and is shown in panel C. Again, we see that rapamycin and the LY compound inhibit S6 phosphorylation. The phospho-ERK blot shows that the UO126 is active and able to inhibit ERK signaling in these cells, but does not affect phospho-S6 levels. The goal of our work that I'm describing to you today was to adapt these low-throughput assays to a format suitable for high-throughput screening. To accomplish this, we turned to the In-Cell Western assay using the Lycor Arius infrared scanner. The In-Cell Western assay is very similar to a standard immunofluorescence protocol using fluorophores in the visible spectrum, with a key difference being that the secondary antibodies used for the In-Cell Western assay are conjugated to specialized fluorophores that fluoresce in the infrared spectrum for detection on the Arius instrument. Specifically, in our assay, cells are plated in a 384-well format, fixed and stained following treatments with a primary antibody against phospho-S6, stained with the IR dye conjugated secondary antibody, and then quantified on the Arius scanner. The Arius has two channels. On the 800 channel, we measure the intensity of the phospho-S6 staining using a phospho-S6 235-236 primary antibody that I described in the previous slide, followed by detection with a goat anti-rabbit IR dye 800 CW antibody from Lycor. The 700 channel is used to measure cell number, which I'll describe in more detail in a moment, and is used to normalize the phospho-S6 signal. The final parameter that we use for our screening assays is the normalized phospho-S6 signal, defined by the intensity of the phospho-S6 staining on the 800 channel, divided by the intensity of a cell number stain measured on the 700 channel. Now I'd like to move on and describe some of the results that we have obtained using the In-Cell Western assay. In the first section of the talk, I'd like to describe the characterization of a number of different cell number stains that we've used for normalizing the phospho-S6 signal as I just described. In previous work, DNA binding dyes that fluoresce in the infrared spectrum, such as TOPRO3, have been used as normalization controls for the In-Cell Western approach. During the early phases of assay development, we found that the intensity of many DNA binding dyes did not correlate well with cell number. These problems were particularly pronounced when cells were transfected with siRNAs that induced apoptotic cell death. An example of this problem is shown on the data presented in this slide. In this experiment, we've transfected cells with siRNAs targeting a number of known components of the mTOR pathway, as well as the PLK1 siRNA, which induces apoptotic cell death, and compared the results of these transfections to two different non-targeting control siRNAs. The cells were fixed and stained with TOPRO3 for visualization on the Lycor Arius instrument, and at the same time were also stained with hooks to visualize nuclei by high-content microscopy. As you can see, siRNA-mediated knockdown of PLK1 induced apoptosis and led to a large reduction in cell number as measured by direct counting of the host-stained nuclei using the microscopy imaging. But this reduction in cell number did not result in a similar reduction in TOPO3 intensity. We speculate that these discrepancies are because the dye bound to fragmented DNA in apoptotic cells has different fluorescence properties to the dyes bound to DNA in intact cells. To identify a more suitable counterstain for cell number normalization, we explored a number of alternative dyes with spectral properties compatible with the 700 nanometer channel on the Arias instrument, including DNA binding dyes such as TOPO3, DRAC5, and Cyto60, an Alexa 680 conjugated phalloidin, which is a far-red F-actin stain, as well as a free Alexa 680 succinimidyl ester, which is a lysine reactive probe that non-specifically labels reactive lysines in the cytoplasm of fixed cells. We also tested additional antibody-based stains for measuring total cell number, visualized with the GOAT anti-mouse IR dye 700 conjugated secondary antibody from Lycor. We first determined whether the intensity of these stains provided a linear measure of cell number. Increasing numbers of cells were plated in a 384 well format, fixed 24 hours later, and co-stained with the candidate normalization dye, as well as 0.5 micrograms per ml hooks to visualize nuclei by high-content microscopy. The intensity of the counterstain was quantified for each cell density using the Lycor area scanner. The number of cells in each well was also measured directly by imaging the hook-stained nuclei on a high-throughput microscope. The results of this analysis are plotted on this slide, showing that each of these counterstains provides a linear measure of cell number over a range of cell densities typically encountered in screening experiments. And in this slide, we're using the raw intensity output from the Lycor Arias instrument. On the next slide, we've performed the same analysis, but in this case, we've used the trimmed mean as the output from the Lycor Arias for our measure of cell number. As you can see in this case, for each of the dyes tested, we see that there's a more linear relationship between cell number and the intensity measured on the Lycor Arias. So for this reason, we've decided to use trimmed mean for all of our subsequent analysis. The trimmed mean simply is excluding the top 20% and bottom 20% of pixels in each well. And I think what's happening here is that we're excluding dust and noise and other bad pixels from the measurement and getting a truer representation of the cell number. The next slide shows a critical experiment where we've calculated the z-factor for an siRNA transfection experiment in which we compared cell number normalization dyes for with a non-targeting control siRNA to an siRNA targeting PTK1. The total S6 antibody provided the most statistically robust normalization for the phospho-S6 signal, with a z-factor of 0.65 in this experiment. However, we deemed the total S6 antibody impractical for high-throughput screening efforts due to the high cost of the second primary antibody. Direct labeling of the fixed cells with an infrared excitable succinimidyl ester dye, the Alexa 680, which directly labels reactive lysine residues and other primary amines in fixed cells, gave a z-factor of 0.59 in this experiment. We concluded that for normalization in a high-throughput screening assay, direct labeling of fixed cells with an infrared excitable succinimidyl ester dye is significantly less expensive, as well as higher throughput, than using separate primary and secondary antibodies to detect the total S6 protein levels. Moreover, it provides a statistically robust measure of cell number, and was therefore used as a cell number dye for the remainder of our screening experiments. The next slide just provides a summary of the conclusions from our analysis of the various cell normalization dyes. As I showed you, the trimmed mean intensity measurement on the Lycor Arias scanner gives a more linear measure of cell number than the total intensity measurement. Protein dyes, including the total S6 antibody and the Alexa 680 succinimidyl ester, are linear with respect to cell number and provide excellent normalization dyes. The DNA dyes DRAC5 and Cyto60 are less effective, but are superior to the TOPO3 we used in our initial experiments. The Alexa 680 NHS ester and the total S6 antibody both gave Z primes greater than 0.5 when comparing the non-targeting control to the PTK1 siRNA, and are therefore provide suitable normalization dyes for high-throughput screening. The Alexa 680 provides an inexpensive and statistically reproducible measure of cell number and was therefore chosen as the normalization dye for the screen. So in the next section of the talk, I'd like to move on and describe some of our small molecule validation data. This next slide gives a summary of the workflow for our 384-well small molecule screening experiments. On day 1, we plate 1,000 HeLa cells per well in 384-well format in 30 microliters of media. The cells are incubated overnight to allow them to become adherent to the plates. 24 hours after plating, we pin transfer 100 nanoliters of compound in DMSO using a pin transfer robot in our screening facility. The compounds are incubated for 2 hours, then the media is aspirated and the cells are fixed with 75 microliters of 4% formaldehyde for 20 minutes. The fixative is washed and the cells are permeabilized with 0.2% Triton X100. We then perform the cytoplasmic staining using the Alexa 680 NHS ester dye as I've described. We again wash the cells to remove the free dye and then block using 75 microliters of the Lycor blocking buffer for 1 hour. We then aspirate the block and add 20 microliters of primary antibody diluted 1 to 1,000 in the Lycor blocking buffer and incubate primary antibody overnight. Following primary antibody incubation, the cells are again washed and then incubated with 20 microliters of the IR dye 800 CW goat anti-rabbit secondary antibody for 1 hour. The cells are then washed 3 times and imaged on the Lycor area scanner. In order to validate the phospho-S6 In-Cell Western assay for small molecule-based high-throughput screening, we first generated dose-response curves for known inhibitors of mTORC1 signaling, including rapamycin and the LY294002 compound. Rapamycin is an mTORC1 inhibitor, and the LY compound inhibits both PI3 kinase and mTOR. The results of the rapamycin dose-response experiment are shown in panel A, with the raw data from the Lycor area scanner shown on the right. The phospho-S6 signal is pseudocolored in green and was collected on the 800 channel, and the cell number data, which is the Alexa 680 NHS ester staining, is pseudocolored in red and was collected on the 700 channel. And as you can see, there's a nice dose-dependent inhibition of phospho-S6 staining. When we quantify this data on the Lycor area scanner, we measure an IC50 value for rapamycin of around 200 picomolar, which is consistent with previously published results. A similar experiment using the LY compound is shown in panel B, where fitting of the data gave an IC50 value of 2.5 micromolar. Importantly, the MEK inhibitor UL126 showed no effect on RPS6 phosphorylation across a range of concentrations where this compound is reported to be active against MEK. Thus, the inhibition that we observe for rapamycin and the LY compound is specifically due to inhibition of the mTORC1 pathway, and the ERK-MAP kinase pathway does not appreciably affect the RPS6 phosphorylation under our assay conditions, as has been shown previously in other systems. To demonstrate the reproducibility of the assay for small molecule screening, we compared treatment of cells with 20 nanograms per ml rapamycin to treatment with a DMSO vehicle control across an entire 384 well plate, as shown on this slide. The image on the left-hand panel shows the raw data output from the Lycor area scanner, which gives you a good sense of what the data looks like coming out of the instrument. I wanted to point out 2 important controls that I include on most of my screening plates. In general, in column 1, I don't plate any cells in order to ensure that there's no background signal from the plates themselves. Similarly, in column 24, I included no primary antibody control, but stain these wells with the secondary antibody to ensure that there's no nonspecific secondary antibody staining. And as you can see, there's very little green signal in those wells, indicating that there's no nonspecific staining. These data were quantified and used to calculate a Z-prime for the assay, and in this particular experiment we measured a Z-prime of 0.80, and typically we measure Z-primes in the range of 0.75 to 0.8 for this assay. These results demonstrate that the In-Cell Western-based phospho-S6 assay is is statistically robust and suitable for small molecule high-throughput screening applications. For those of you not familiar with Z-prime calculation, this is essentially a measure of the statistical reproducibility of the assay, and the equation on the bottom panel shows the calculation. A perfect assay will give you, which is one that has no variation at all, will give you a Z-prime of 1.0, and Z-primes between 0.5 to 1 are typically considered suitable for high-throughput screening. So again, as you can see, the Z-prime of 0.8 that we measure for the In-Cell Western assay is indicative of a highly reproducible assay that's suitable for high-throughput screening. In the next slide, we show a similar experiment performed using high-content image analysis. Again, comparing rapamycin treatment to DMSO control across an entire 384-well plate. In this experiment, the cells were stained with hooks to visualize nuclei, and also stained with the phospho-S6, which was visualized in this case with a goat anti-rabbit Alexa 488 antibody, secondary antibody for visualization on a high-content microscope. The data was analyzed by first identifying nuclei using computer-assisted image analysis. We then defined a cytoplasmic area as a ring around that nucleus and quantitated the phospho-S6 staining on a per-cell basis. The statistical analysis of this data is shown on the next slide, where we've calculated a z-factor for this experiment. And in this particular experiment, we've measured a z-factor of 0.60, and typically for high-content microscopy-based quantitation of the phospho-S6 assay, we measure z-factors in the range of 0.58 to 0.65. I should note, for those of you that are high-content imaging experts, that we've done more sophisticated image analysis using water shedding algorithms and cytoplasmic stains to really very carefully define the cell area, as well as including localized background corrections. And even with these more sophisticated image analysis approaches, we can only increase the z-prime up to about a limit of 0.65. And so consistently we find that the In-Cell Western assay is superior with regard to statistical reproducibility to similar assays performed by high-content microscopy. And the next slide just shows a summary of our comparison of the In-Cell Western assay and high-content immunofluorescence-based assays for measuring, for quantitating S6 phosphorylation for high-throughput screening. In the In-Cell Western assay, we were able to use the phospho-S6 primary antibody at a 1 to 1,000 dilution. In contrast, in order to obtain sufficiently high signal intensity to make the exposure time short in an immunofluorescence-based assay, we need to use slightly higher antibody concentrations, ranging from 1 to 100 to 1 to 500 dilutions. In the In-Cell Western assay, we have a scan time of about 5 minutes, whereas in an immunofluorescence-based assay, to scan an entire 384-well plate takes about 2 hours. On the In-Cell Western assay, the data is actually analyzed on the fly during the scan, but we estimate it takes about 10 minutes to export this data into Excel and perform some manipulations in order to view the data. Whereas analysis of high-content images using computer-assisted image analysis will typically take 2 to 3 hours. And finally, the In-Cell Western consistently shows a superior Z-prime in our hands in the range of 0.8, whereas immunofluorescence-based assays are giving us a Z-prime of 0.6 to 0.65. So moving on to some screen data, following validation of the phospho-S6 In-Cell Western assay, we performed a pilot small molecule screen using a library of 2,640 known bioactive molecules available through our screening facility here at the medical school. Each library plate was screened in duplicate, and the average Z-score for both cell viability using the Alexa 680 NHS-ESSER staining and the normalized phospho-S6 staining was calculated. The results of this screen are summarized in the panel on the top of this slide, which plots the average Z-score measured for the phospho-S6 assay for each compound in the known bioactives library. In the pilot screen, cytotoxicity was defined as a reduction in the Alexa 680 6-phenyl ester cell number staining greater than 3 standard deviations relative to the plate median. That's a z-score of less than -3. 27 compounds, or 1% of this known bioactives library, scored as cytotoxic on both replicate plates in this pilot screen. And these are indicated by the yellow stars in the upper panel. In the next slide, we show a summary of the results of the In-Cell Western assay and high-content immunofluorescence-based assays for measuring S6 phosphorylation for high-throughput screening. In the In-Cell Western assay, we were able to use the phospho-S6 primary antibody at a 1 to 1,000 dilution. In order to increase the Inhibition of the RPS6 phosphorylation was defined as a reduction in normalized phospho-S6 staining of greater than 3 standard deviations relative to the plate mean, again a z-score of below -3, as indicated by the red line. 18 compounds, or 6.8% of the library, exhibited a z-score of less than -3 for the phospho-S6 assay on both replicate plates without significantly affecting cell viability. And these are indicated with the red symbols. An additional 4 compounds scored with a phospho-S6 z-score of less than -3, but were excluded as hits because these compounds also scored as cytotoxic. And one thing that I'd like you to note is that the cytotoxic compounds are sort of distributed throughout the plot, and they're not really clustered in any particular location, indicating that we're doing a very good job of normalizing for the phospho-S6 signal, and there's no bias introduced by changes in cell number. Importantly, known inhibitors of the mTORC1 pathway, including the pan-kinase inhibitor sporin that's known to inhibit S6 kinase, the mTORC1 inhibitor rapamycin, and the dual PI3 kinase and mTOR inhibitor LY294002, all scored among the most potent inhibitors of the pathway and are indicated with the arrows and green symbols on the plot. These results validate the ability of our method to identify known inhibitors of the mTORC1 pathway in an unbiased screen. The reproducibility of the In-Cell Western assay in this pilot screen is demonstrated in the data on the lower panel of this figure, which plots the z-score for the phospho-S6 staining from two independent replicates of a representative plate from the small molecule library. A linear fit of this data gives an R-squared of 0.91, which is indicative of an excellent plate-to-plate reproducibility within the assay. So we were very satisfied with the results of this small molecule screening effort, and we went on to validate the In-Cell Western assay for siRNA screening. The results of that analysis will make up the third and final section of the work that I'm going to describe to you today. This next slide just summarizes the workflow for our siRNA screening experiments. On the first day of the experiment, we perform a reverse transfection of HeLa cells in a 384-well format using 50 nanomolar siRNA final. And this primary screen was performed with Dharmacon smart pools, which contain 4 individual siRNAs as a pool targeting each gene. 24 hours after the transfection, the cells are fed with 55 microliters of complete media containing antibiotics and serum. The cells are then incubated for 48 hours to allow for knockdown of the target genes. 72 hours after the initial transfection, we aspirate the media and fix the cells with 4% formaldehyde, and then process them for quantitation on the Lycor Arius instrument using a protocol identical to that I've described previously for the small molecule screen. The data on the next slide shows the results of an experiment that we performed in order to validate the phospho-S6 In-Cell Western assay for siRNA-based high-throughput screening. In this experiment, we tested the ability of siRNA-mediated knockdown of known components of the mTORC1 pathway to block the phospho-S6 signal. Using the reverse transfection protocol that I described in the previous slide, we transfected a non-targeting control siRNA, as well as siRNA pools targeting the indicated components of the mTORC1 pathway, as well as an siRNA pool targeting PLK1, which induces apoptotic cell death, as I described earlier. The raw image from the area scanner is shown on the left-hand side of this panel, and again, the phospho-S6 stain collected on the 800 channel is shown in green, and the cell number stain, the Alexa 680 NHS ester, is pseudocolored in red and was collected on the 700 channel. As you can see, both the PDK1 siRNA and a pool of S6 kinase 1 and S6 kinase 2 siRNAs led to a dramatic reduction in the phospho-S6 signal. The quantitation of the phospho-S6 staining is shown on the upper right panel on this slide, demonstrating a greater than 80% reduction in the phospho-S6 staining in response to knockdown of either PDK1 or both S6 kinase isoforms. Knockdown of S6 kinase 1 or S6 kinase 2 individually results in a more modest decrease in S6 phosphorylation, demonstrating that both isoforms of this important kinase contribute to S6 phosphorylation in HeLa cells, with S6 kinase 2 apparently playing a more significant role. Finally, an siRNA pool targeting the mTOR kinase leads to a relatively moderate 40% reduction in S6 phosphorylation, consistent with the incomplete knockdown of mTOR protein achieved by this siRNA, which we've confirmed in other experiments. I also wanted to point out the quantitation of the cell number data shown in the lower right panel on this slide, where we see a greater than 80% reduction in the cell number in response to apoptotic cell death induced by the PLK1 knockdown. This was nice confirmation that the Alexa 680 NHS ester staining provides a good normalization for cell number, even under conditions of apoptotic cell death, and avoids the problems that I described to you earlier in the beginning of the presentation. In the next slide, we performed an experiment to demonstrate the reproducibility of the assay for siRNA screening in a 384-well format. In this experiment, we compared knockdown of the S6 kinase 1 and S6 kinase 2 siRNAs to transfection of a non-targeting control siRNA across an entire 384-well plate. Again, the raw image is shown on the left, and you can see by eye that we get very consistent inhibition of S6 phosphorylation across the entire plate. On the right-hand panel, we show the quantitation of this data that was used to calculate a z-factor for the experiment. And we typically measure z primes between 0.67 and 0.65 in this siRNA transfection experiment. Again, demonstrating that the In-Cell Western-based phospho-6 assay is statistically robust and suitable for high-throughput siRNA screening applications. Following validation of the phospho-6 assay for siRNA screening, we went ahead to perform a pilot screen using a library of 779 siRNA pools targeting all of the known protein kinases and protein phosphatases in the genome. And the results of this screen are shown on the next slide. On the left-hand panel, we've plotted the average z-score calculated from the phospho-6 assay for each siRNA in the screen and arranged these in order of hit strength. siRNAs that inhibit the phospho-6 assay were defined as those that result in a change in the normalized phospho-6 staining of minus 2 standard deviations relative to the plate median on both replicate plates. Hits in the assay are indicated by red symbols on this plot. Using these criteria, 48 genes, or 6.2% of the kinome and phosphatome, score as inhibitors in this screen and are expected to target genes required for S6 phosphorylation. Importantly, siRNAs targeting known regulators of mTORC1 signaling, including PDK1, S6 kinase 2, AKT2, and the IGF-1 receptor, scored among the most potent inhibitors of the pathway in this pilot screen, validating the ability of this method to identify known components of the mTORC1 pathway in an unbiased screen. An additional 12 genes, or 1.5% of the library, led to an increase in S6 phosphorylation and are expected to target genes that negatively regulate S6 phosphorylation. Similar to the data that I described for the small molecule screen, cytotoxic siRNAs were defined as targets with a reduction in cell number of more than 3 standard deviations below the plate median on both replicate plates, and are indicated with the yellow symbols. 20 genes, or 2.6% of the kinases and phosphatases in the library, were required for cell viability as measured by a statistically significant reduction in the Alexa 680 NHS ester staining. To validate the results of the primary screen, we performed an experiment in which we went back and retested each of the 4 individual siRNAs that comprised the SMART pool that scored in the primary screen. The results of this analysis are shown in panel B on this slide. As you can see, 36% of the hits that were scored in the primary screen retested with at least 2 out of 4 of the individual siRNAs, indicating that these targets are of high confidence and likely represent novel regulators of the mTORC1 pathway. We are currently working to characterize these hits and hope to publish our results soon. That concludes the data portion of the talk. Before starting the question and answer session, I'd just like to acknowledge some of the people that helped me with the screen. In the Blenish lab, Max Shaw was a technician who was really critical in helping to develop the In-Cell Western assay in the early going. In the screening facility here, the Institutes of Chemistry and Chemical Biology, or the ICCB, I've already mentioned that the project was a collaboration with Carolyn Chamoux and Nate Moricky. We've also received help from Stuart Rudnicki and Sean Johnstone, who are staff members in the ICCB that assisted with many of the siRNA transfection experiments. Finally, I need to thank Pete Johnson at Lycor, whose help in the early stages of the assay development was really instrumental in getting this project off the ground. Thanks again for listening to the presentation, and now I'd like to take any questions that you might have about the assay.
All right, I think we're ready to go now. Greg, our first participant asks, can you explain why you chose to use the areas for your screen instead of other similar technologies, such as a microscopy or plate reader-based approach?
Yeah, that's a good question. I mean, I touched on some of this in the presentation. First of all, plate reader approaches, we actually tried using a plate reader for the assay, and we simply didn't get enough signal off of a plate reader. I think it's really important to have either a microscopy or a laser scanning instrument like the Arius to focus the light and really only collect information on the cells that are fixed in the bottom of the plate. In terms of the comparison with a microscopy-based assay, really one of the primary considerations for us was the cost. In order to get significant, reasonable throughput with a microscopy-based assay, we had to use higher antibody concentrations, which significantly increased the cost of our primary screen. In addition, for this assay, we're really only interested in yes or no kind of information, whether S6 phosphorylation is on or off in a particular condition. And there really isn't a lot of additional information in the high content image from a microscope. And so in that regard, a high content screening approach really requires a lot of extra labor and a lot of extra time without really adding a lot of new information to the experiment. And so for that reason, we were really happy with the laser scanning instrument with the Arius, because it gave us a really high throughput without losing any information. I hope that answers your question.
Great, thank you, Greg. Our next participant asks, what is the throughput for your assay when you are doing a large-scale screening project? What are the bottlenecks?
Yeah, that's a really important issue when you're doing these large-scale experiments. In general, when we're doing an RNAi screening project, our throughput is about 20 plates per week. As you can see by the workflow that I outlined earlier, it's a fairly complicated experiment, and really the primary bottleneck for us is the transfection step. So we can reasonably handle about 20 plates at a time for a transfection. So in general, we're doing this in duplicate, so that would be 10 separate siRNA plates that we're able to screen and duplicate in a week. The Arius, the quantitation of the plates is really very fast. So again, we're scanning each plate 5 to 10 minutes. So for 20 plates, that gives us, what's that? I should do this in my head. Roughly an hour and a half to 3 hours to scan a stack of 20 plates. So that's really very reasonable. For small molecule screening experiments, I've never done more than 20 plates in an experiment, but there's not this transfection bottleneck, and I think it would be reasonable to screen 40 or 50 plates a week very easily.
Great, thank you. Do you have a preference for pooled versus individual siRNA libraries?
Yeah, the screening facility here at the ICCP has settled on a pooled approach, and this is really for practical reasons. The pool approach dramatically reduces the number of libraries the number of library plates that you have to screen to cover the entire genome. So if you're screening an unpooled library with 4 individual SIs targeting each gene, that would increase the size of the primary screen and also increase the cost of the primary screen 4-fold. And we found that the strategy of screening pools and then going back and retesting the individual siRNAs as a secondary screen is a good strategy, and I don't think we miss a lot of individual SIs that we would not pick up in using an unpooled library. So I think the benefits in terms of cost and throughput, particularly for an academic institution, are really dramatic for the pooled library.
Great, thank you. Our next participant asks, if I'd like to develop an ICW assay for my favorite protein, do you have any suggestions for how to optimize and troubleshoot a new antibody?
Yeah, I mean, again, the In-Cell Western assay you should really think of as very similar to a standard immunofluorescence protocol. So if you have an antibody against your favorite protein that already works in an immunofluorescence-based assay, that will typically transfer very well to the In-Cell Western. If you're talking about piloting a new protein, I think the first thing to do is just want to run a Western and make sure that the antibody is recognized recognizing a single species. And once you're confident that you have a pretty good antibody, perform essentially the same kinds of normalization, the same kinds of optimization experiments that you would perform in an immunofluorescence assay. If I have an siRNA targeting the gene of interest, I like to knock down the si and show that the signal goes, or knock down the protein with the si and show that the signal goes away when I knock down the protein. And obviously, if you have a positive control, it will change the expression level or the phosphorylation status or whatever is being detected by the antibody. Those are important as well. And always include a no primary control to make sure you're not getting any background staining, although I've never had any problems with that.
All right. Thanks, Greg. I think this is going to have to be our last live question, and I want to thank the audience for their submissions. And I also want to remind the audience that if they haven't yet submitted a question or they have submitted a question and it hasn't yet been answered, or Greg didn't get to it during the live session, to please stick around and a panelist will get back to you as soon as they can.